Thanks, David. Yeah, it's interesting that the cell-free protein synthesis field has been around for decades, even more than a century. The idea that you could take living organisms, rip off their cell walls, collect the insides, and then try to gain insights into how biology is working, but also use that for perhaps making products. Has certainly been around for a long time. Of course, we have the very early experiments when we just through the discovery of the genetic code in the 60s, and that of course developed into many fundamental insights that were garnered about how the processes of transcription and translation, the way in which the flow of information through the central dogma of biology can be controlled and captured and regulated. And then, as time moved on, there began to be questions about, well, that's very powerful. Can we use this approach, this cell-free based approach, not just to understand, but maybe to make enzymes, to make proteins that then we can study as a secondary element? And really, I think there were some transformative experiments, one early on by Spearin's group, and then much of the work that came out of the Schwartz lab at Stanford University trying to develop the cell-free protein synthesis systems as a platform for manufacturing proteins that could be used, studied, or potentially even leveraged as therapeutics themselves. And so I think very interestingly, kind of in the mid to late 90s through the early 2000s, there were many innovations that transformed cell-free protein synthesis systems into a potential technology for making therapeutics. If you look back to the late 1990s and early 2000s, there were a whole lot of challenges around cell-free protein synthesis systems. They were very short reaction durations. So the reactions might last 5 minutes, 10 minutes, 15 minutes, if you're lucky. The systems were very expensive. You used high energy phosphate compounds as a secondary energy substrate to replenish ATP, which is required for protein synthesis. So you can imagine molecules like phosphoenolpyruvate, which cost hundreds of dollars per gram, rather than inexpensive substrates like glucose, which are used to grow cells. The system really hadn't been demonstrated to produce complex protein therapeutics or proteins with disulfide bonds, for example. And so really there were many challenges surrounding the use of cell-free protein synthesis systems. And kind of one by one, really by focusing on the substrates rather than the products, focusing on the stability of the RNA and DNA template, focusing on the stability of the amino acid substrates, the energy supply systems, etc. The field was able to continue to increase protein synthesis yields, extend batch reaction durations, and reduce costs, all the while being able to make more and more complex proteins. And so really there's this transformative period in the development of the cell-free protein synthesis field. While costs are being reduced, protein synthesis yields are increasing, and the complexity of the type of proteins that you're making is increasing. And fast forward 10 years, we're here, right? I think that those early innovations really led to the formation of, for example, Suture Biopharma, which was a company that spun out of work in Jim Schwartz's lab at Stanford University, really focused on making protein therapeutics, now looking in one example at antibody drug conjugates or others. And it really began to make possible this idea that you could use cell-free systems to manufacture products. Some other key innovations that occurred in the field just over the course of the last decade is now this an additional transformation to not only being able to synthesize proteins at high yields, at lower costs, address the scale-up issues. So learning how to, for example, carry out cell-free protein synthesis reactions at not just the nanoliter, microliter scales, but really the 10, 100, and perhaps even 1,000-liter scales. But in addition, the determination and realization that these cell-free systems could be freeze-dried and maintain activity through some of the early work of Brad Bundy, Jim Collins, some of the work coming out of our own group and others, really developing strategies to kind of freeze dry these cell-free systems and demonstrate that they could be used as a portable manufacturing platform for on-demand synthesis has really, I think, accelerated a lot of interest in using cell-free systems, especially for manufacturing medicines, therapeutics, chemicals, or other molecules when and where we need them.